Good afternoon, everyone. Welcome to our webinar about ICDs and immunotherapy. It's 5 o'clock in Europe and 11 o'clock on the East Coast of the US. We are getting started on time. Today's webinar is called How to Kickstart the Immune Response in Anti-Tumor Therapy, a unique screening platform to identify immunogenic cell death inducer. Before we begin, I'd like to address a few practical matters. First, we are recording this webinar and the presentation will be available to all attendees afterwards. Then, after the presentation, Akanksha will answer a few of your questions. So if you have any, please feel free to type them in the question panel. We will gather all the questions at the end of the presentation and Akanksha will answer them to you. Also, if you're experiencing any technical difficulties, please directly ask in the chat. We'll do all we can to help you out. Finally, and before we start, I am opening a short poll question. Please take a second to answer. We'll be happy to assess your level and to adjust our webinar according to the knowledge of our attendees today. So I'm looking forward to receiving your answers on the poll. While you're answering, I will introduce you Dr. Akanksha Ganga. So Akanksha obtained her PhD in biochemistry in the prestigious Indian Institute of Science. She then continued as a research associate at the University of North Carolina to study cell polarity. In 2005, Akanksha worked at INSERM as a research scientist to work on the cell polarity in an oncology context. In 2007, she joined GSK as their R&D at the R&D center located in France to support the drug development in hypertension, obesity, and diabetes. Since 2016, Akanksha has been working in the in vitro department of Oncodesign as a scientific expert and a study director. Our focus is on cancer-related projects. Akanksha, are you ready to start the webinar, the presentation?
Yes, Krista, thank you.
All right, so I'll let you begin.
Hello, everyone. It's great to have you on board for this webinar on ICD. So today's key learning objectives will be introduction to ICD, how Oncodesign can help you identify and characterize ICD inducers, and I'll present some case studies. So how did all this start? Well, on a foggy and rainy autumn day, we got a call from a sponsor. And he asked us if we had assays in place to identify ICD inducers. And we said, yes, what exactly are you looking for? And he said, well, I want to screen a library of about 10,000 molecules, and I would like to identify ICD inducers. He said, well, we can surely help you in getting that done. How much time we have to set base and get everything ready? And the sponsor said 3 months. We said, okay, great. And as soon as we put the phone down, we went to the literature and we first tried to get on buff about ICD. He said, so what exactly is ICD? For many years, we know that apoptosis has been considered as a non-immunogenic process, whereas necrosis is acknowledged to play a key role in inflammation. A new concept has emerged since a couple of years, which is called immunogenic cell death, or ICD. And this has challenged the traditional view and has granted apoptosis with immunogenic abilities. This paradigm shift offers clear implications in designing novel anti-cancer therapeutic approaches. So, in response to immunogenic cell death signals, malignant cells emit danger signals. secreted ATP is one of them. It is a potent short-range find me signal that can bind to purigenic receptors. Secreted ATP also has been observed to help in dendritic cell activation. Another danger signal is calreticulin. So calreticulin normally is an endoplasmic protein, and upon endoplasmic stress, it is translocated from the endoplasmic reticulum lumen to the outer leaflet of plasma membrane. And it functions as an eat me signal, thereby assisting in the phagocytic uptake of dying cancer cells. Moreover, the calreticulin at the membrane is capable of inciting the production of IL-6 and tumor necrosis factor from dendritic cells, thereby facilitating Th17 polarization. Another signal is the release of HMGB1, or high mobility group protein B1. This protein then binds to the TLR receptor on dendritic cells, and on one hand activates production of pro-inflammatory cytokines, and on the other hand assists in proper antigen presentation. Interestingly, it has also been found to suppress the activity of immunosuppressive Treg cells. So, upon binding of the cognate receptors to the surface of myeloid or lymphoid cells, these danger signals favor the uptake of dying cancer cells or debris, thereby presenting them to dendritic cells in the context of robust immunostimulatory signals, eventually leading to the priming of an adaptive immune response, as we can see over here, involving both gamma delta and alpha beta T cells. In addition to the establishment of immunological memory, such a response also has the potential to eradicate malignant cells that survive chemotherapy via an interferon gamma dependent mechanism. So, ICD relies on the establishment of stress responses that promote the spatiotemporally coordinated emission of endogenous danger signals from dying cells. There are three categories of danger signals or DAMs. The surface exposure of chaperones. This is pre-apoptotic. Secreted or released nucleotides or nucleic acids. And the release of endogenous toll-like receptor agonists, which is mostly mid-apoptotic stage. A simple structure-function relationship for ICD inducers is virtually non-existent and perhaps impossible. However, a classification scheme for all the ICD inducers has been proposed. This scheme is based on the ability of an ICD inducer to target the ER and to induce as many ICD-relevant processes as possible. Type 1 ICD inducers are modalities that induce cell death via non-ER associated targets and danger signaling via ER stress. Whereas type 2 ICD inducers selectively target the ER to induce both cell death as well as danger signaling. Several anti-cancer agents have been successfully employed in the clinic for decades, such as oxaliplatin, etc., and have been found to elicit active immunological anti-tumor response via ICD. However, if the chemotherapeutic agent fails to induce one or more of these DAMs, then the ICD response is compromised. So we learn from the literature that we need at least 3 different markers to have complete ICD. There are several molecules that are gold standards and well known in the literature. So we set about to define the strategy to set up what the sponsor wanted. So the objective was to develop a new and a unique screening platform to identify immunogenic cell death inducers. We had several steps to go forward in doing so. The first one was to determine the lowest toxic dose. As you would have seen in the previous two slides, that ICD finally ends up in cell death via apoptosis. So we don't want to go as far as generating apoptosis, and hence we need to identify the lowest toxic dose. The second step would be to identify the compounds that secrete ATP. This is an easy marker to look at and would give us the first indication on ICD inducer. And all of this in the absence of toxicity. Finally, we need to make sure that all the three ICD markers are indeed present upon treatment with the ICD inducer. ICD doesn't end over there. We need to make sure that the ICD inducer is able to activate dendritic cells, which in turn would activate the T cells, and that would be a step 4 and step 5. So, mitozentrone, a well-known ICD inducer in the literature, was considered to be used as one of the positive controls. Another positive control that we decided to use came from a publication, a recent one in 2018, called Nanocyclix. This one is a CDK inhibitor, and as a company, since we are interested in kinase inhibitors, this was of interest to us. We tested these compounds over different time points in as a mouse cell line, CT26, which is a colon carcinoma from mouse bulbs. So the first step was to identify compounds that with lowest toxic dose, and then that lead to secretion of ATP. To do so, we used two different assays. To look at intracellular ATP, we use the cell titer glow assay, whereas to look at extracellular ATP, we use the Enlighten assay. When we look at the results that we obtained for the intracellular ATP at different concentrations of mitozentrone or dinaciclib, at 24 hours, we see nearly no toxicity, with some toxicity observed at 48 hours. The cutoff that we established was cell viability of over 70%. And that's why you see 65% or 61 and 36 in red, because they indicate toxicity. And that's the color code that we'll be using throughout this presentation. At the same time points and the doses, when we looked at extracellular ATP, so secreted ATP, we found that mitozentrone resulted in some secretion of extracellular ATP, both at 24 and 48 hours. And dinaciclib was more potent because we had a lot of doses that resulted in the secretion of ATP. Anything above 150% over DMSO was considered as positive and represented here in green. So what we chose was dinaciclib, because that's a compound that resulted in a nice secretion of ATP without toxicity. The next step was to look at the different ICD inducers, namely HMGB1 that we looked at via ELISA, and calreticulin via FACS or immunofluorescence, depending on the cell line that we choose. So the first one in line, HMGB1. When we look at dinaciclib and the secretion of HMGB1 in the extracellular medium, at 24 hours and at 44 hours, we see a nice secretion of this protein. Calreticulin also is translocated from the ER to the outer leaflet of the plasma membrane at the same doses and at 24 hours. So we've identified dinaciclib as an ICD inducers, which results in all the three ICD markers: extracellular ATP, HMGB1, and calreticulin, and all of that at non-toxic concentrations in CT26 cell line. Now that we've identified dinaciclib, the next question that we asked was, is this enough to result in the activation of DCs, which is the first player in the adaptive immune response? To do so, we used the following strategy. On day 1, we labeled CT26 cells with vibrant DIO. The next day, we treated them with Nanocyclix. As we know from the previous slides and our in vitro assays, that 24 hours was sufficient to elicit an ICD response. We let them in Nanocyclix for 24 hours, following which we added mouse DCs, dendritic cells. And 24 hours later, we labeled them and looked at via FACS or performed cytokine analysis via multiplex. In the graph on the left, we can see the FACS analysis where we looked at the marker DIO, which is a marker for CT26 cells, and CD11C, which is a marker for dendritic cells. When there's double positive cells, we consider that as phagocytosis of CT26 cells via the DCs. And what we observed was that at 0.1 and 0.2 micromolar Nanocyclix, there was an increase in the double positive cells. At the same doses, we can see a nice increase in the cytokines that we looked at, IL-1 beta and IL-6, the graph on the right. So what we were able to demonstrate is that the CT26 treated with Nanocyclix, when co-cultured with dendritic cells, were taken up and phagocytosed by the DCs. So that's what is represented here in this little picture. We have double positive cells, and these cells secrete IL-1 beta, IL-6, IL-12, and TNF-alpha. Having seen this, we were really curious to find out if the activated DCs in turn stimulated T cells. So to do so, we used almost the same scheme as before. However, on day 4, instead of labeling the cells, we added mouse T cells, which were CFSE labeled to follow their proliferation. After several days of incubation, we looked at the T cell population via FACS and performed cytokine analysis via multiplex. On the graph on the left, you can see CFSC CD4 or CFSC CD8 positive cells. So in blue, it's the CD4 positive cells, and in green, the CD8 plus positive cells. At the doses that we had seen before for the activation of dendritic cells, we can see also a polarization of CD plus cells, where we can see an increase in the CD8 plus population at 0.1 and 0.2 micromolar Nanocyclix. At the same concentrations, we can see an increase in interferon gamma, a key cytokine, IL-6, and IL-17. So what we could conclude from these experiments was that T cells were activated and polarized in favor of CD8+. The DCs loaded with Nanocyclix treated CT26 cells efficiently were able to stimulate tumor-specific interferon gamma producing T cells. Cross-priming of CD8+ cytotoxic T lymphocytes seemed to trigger and mature DCs and gamma delta T cells in an IL-1 beta and IL-17 dependent manner. So to summarize what we had set up is as follows. We had set up a screening strategy to identify ICD inducers. The 3 molecules that we followed to determine if they were ICD inducers was HMGB1, ATP, and calreticulin. We also validated other ICD readouts like HSP90 by immunofluorescence of FACS, caspase-37. To look at ER stress, which is one of the key factors in ICD, we looked at PERK, EIF2 alpha, and its phosphorylation, and reactive oxygen species. When we co-culture dendritic cells and cytotoxin-treated cancer cells, we observe that the dendritic cells get activated and phagocytose the dying cancer cells, secrete cytokines, which then in turn activates T cell and favors its polarization to CD8 plus cells and the secretion of interferon gamma, IL-1 beta, and IL-17. So we looked at not just CT26, but a whole bunch of different human and mouse cell lines, and we identified different molecules that could serve as controls, as positive controls for the different ICD readouts. And we validated, as you can see in this slide, different cell lines. Now that we were ready with the whole ICD screening strategy, we went back to the sponsor and we said, hey, we've got the whole setup and we are now ready to screen your library. And there we were. We got over 10,000 compounds to screen, and our goal was to identify ICD inducers. So I can't, of course, show you the sponsor's data, but however, what I'll show you is the screening of our own internal Oncodesign compound library called the Nanocyclix compound library. The Nanocyclix is a chemistry technology that is based on the microcyclization of small molecule hinge binders of kinase ATP active site. This leads to low molecular weight kinase inhibitors with unique binding mode. With a unique mode of action, they are highly potent and selective. So we followed the same strategy as I described before. Determine the lowest toxic dose. We can do that in a 384 well plate format. We did that for our library using 6 different cell lines, 3 human and 3 mouse, over different time points to look at the cell viability. Once we've identified the lowest toxic dose, we use at least 5 different concentrations to look at the next readout, which is secretion of ATP. This was performed in a 96 well format, once again using all the 6 cell lines over different time points. And then finally, we look at the 2 remaining parameters, which is HMGB1 secretion and calreticulin at the plasma membrane. This is done in the 96 well format. And usually we choose between 5 to 8 different concentrations to identify the potency of the inducer. So as mentioned before, we've successfully screened a sponsor's library of over 10,000 compounds and Nanocyclix compound library. So the results from the Nanocyclic compound library are here. We screened over 2,000 compounds. After identification of lowest toxic dose, we were left with 144 hits. Secreted ATP was observed for 24 hits. And then finally, at the end of step 3, we found an ICD inducer, ODS 336. So quickly to show you what the data looks like for the ICD screen, the ODS 336, you can see here with 3 different cell lines in the first graph, and then the second 3 other cell lines at different time points leads to secretion of ATP. HMGB1 is released at 48 hours from 5 of the 6 different cell lines. Calreticulin is detected either by immunofluorescence or FACS, once again, depending on the cell line, at different time points in different cell lines, either by FACS or by immunofluorescence at the membrane over here. And each time when we have an increase of over 150% over DMSO, we consider that as positive, highlighted here in green. So we are very happy to find a kinase inhibitor that resulted in an increase in these different signals for ICD. So we went further and we said, okay, that's great. It can show the release of HMGB1, cell reticulin at the surface, and secretion of ATP. What about activation of dendritic cells? So we went ahead to perform the co-culture experiment, and we found that there was a dose-dependent increase in the double positive cells that were treated with ODS 336. And at the same concentrations, we could see a nice increase also in the cytokines IL-6 and interleukin-1 beta. So ODS 336 treated CT26 cells, enhanced DC function. We've also been able to set this model up in the HEPA-16 C57 Black 6 mouse dendritic cell model. Once again, we got a call from another sponsor, and they asked us, well, you guys have the mouse co-culture assays. What about the human assays? And we said, that's definitely not a problem. We can set that up. And that's exactly what's depicted here. The human phagocytosis assay, where from human blood, we isolate monocytes, let them differentiate into dendritic cells, and then put them in co-culture with MDAMB 436 cells that are labeled with DIU. We seed them, treat them just like we do for the mouse co-cultures, as you've seen before. And then we look at double positive cells. We see that the doxorubicin treatment of MDAMB 436 cells results in the activation of DCs, as you can see here, with increasing concentration, we see an increase in double positive cells. We are at the moment performing the cytokine profiling to make sure that indeed there is secretion of the cytokines of interest. And of course, the last and the final step is, does this work in the human setup where we can look at T cell activation upon dendritic cell activation? So those are the assays that are ongoing. So in conclusion, what I've shown you, and I hope I've been able to convince you, is that we've established a general strategy for the identification of ICD inducers within large chemical libraries. Using our platform as a case study, I hope I've been able to convince you that we've identified ODS 336 as an ICD inducer, a compound that elicits in vitro ICD response. We have secreted ATP, HMGB1 release, and surface calreticulin. Also, we see DC activation. We've also set up ex vivo co-culture assays for both mouse and human for the DC cell function, and for the T cell activation, the mouse assay, the human assay is in progress, suggesting that the ICD activates both innate and adaptive arms of the immune system. So, where can Oncodesign come and help you in the entire drug discovery process? We can start from the screening. We have the capacity to screen for a medium throughput screening or lower. We can validate your compounds of choice, either for repositioning or for identifying ICD inducers. And then we have in place in vivo mouse models. So Oncodesign can support your drug discovery efforts either on a standalone basis or on an integrated basis. Just to give you a brief glimpse of what we can do and what are the gold standards for in vivo mouse models in ICD. The gold standard for ICD in vivo is as follows. The in vivo vaccination strategy. So the first experiment that can be conducted is inject the cytotoxicin treated tumor cells in immunocompetent mice and see if there is tumor growth or not. If the ICD inducers is a successful one, then there should be no tumor growth. The real experiment is the vaccination experiment, because we said that ICD is involved in memory and in innate and adaptive arms of the immune system. So we would take this mice over here and inject it now with the live tumor cells, the same ones that were used over here. If the ICD inducer is a real ICD inducer, then there would be no tumor growth. However, if it was permissive, there would be tumor growth. So what we can see is, for an inducer to be qualified as a good ICD inducer, we would need to see that in the vaccination in vivo experiment, there's no tumor growth. And these are the experiments that Oncodesign has the capacity to perform. So we can lead you from screening all the way to lead optimization and candidate selection in an integrated manner. So once again, Oncodesign can support your drug discovery efforts on a standalone or integrated basis. Thank you so much.
Thank you very much, Akanksha, for your presentation. So it's the Q&A time. You are very welcome to ask all the questions you may have in the chat panel, and Akanksha will be happy to answer them to you. Akanksha, I received one question already, which is the following one. What is the added value of your platform?
That's a nice question, because of course a lot of companies offer these services. Our added value is that we have an ICD platform where we offer all the assays for all the three ICD markers: secreted ATP, calreticulin at the surface, and HMGB1 secretion. This is very important, because if we miss one, then we may not have a true ICD inducer. In addition, we can perform in vitro co-culture experiments, which would add strength to going forward into in vivo setting. And of course, as I mentioned, Oncodesign has the capacity to perform the in vivo experiments as well. So our strength is to start at the very beginning and end in a mouse model where we can say, yes, you do have a real good ICD inducer from the beginning to the end, an end-to-end solution.
Okay, thank you, Akanksha. I was reading the following question that came up. So I need a second because I can't read it all. It says, "Dear Akanksha, how long it took to screen the 10,000 compound library of the," and that's where it's hard for me to read the following. I have a hard time. I can't see the full questions.
It's okay. I can answer you. Maybe they come up with yeah. To perform the first two steps, it's pretty straightforward. For the 10,000 compounds, it took us less than a month. Once the sponsor had identified a short list of compounds that they wanted to proceed for the HMGB1 secretion and calreticulin, it all depends on the number of compounds that you choose. These assays, as you can imagine, are long, depending on how long is the incubation time, either 24 to 72 hours. So it can take several weeks to finish up the screening. But all in all, for the 10,000 screen, we took about 4 to 4.5 months to screen the entire compound library and to come up with a short list of ICD inducers that resulted in all the three markers.
Okay, thank you, Akansha. So I went to the end of the questions. So there was more than one question there was several questions in one. The rest of it was did you screen one concentration or more, and the compound was dissolved in DMSO?
Yes, the compound library, both our library and the Nanocyclix compound library, being small molecule compound libraries, were dissolved in DMSO. We've tested other compounds that were dissolved in PBS. That's possible. We tested, usually when we do the screen, we test several different concentrations. So for each compound, we test between 5 and 8 concentrations. And the first 2 steps usually give us an idea of what time point we would choose for the next 2 readouts, which are long and time consuming to look at.
All right, thank you, Akansha. There is more questions coming up. The following one is, do you know whether ICD's inducer are at clinical development stage now?
Yes, there are several in clinical development, several in co-development with other molecules like the checkpoint inhibitors. And you can, I think on my second slide, you have a glimpse of some of the inducers and the potential outcome in clinic and for which cancers they've been used.
All right. Next question is the following one. Have you done RNA expression to try to identify an expression signature of ICD? Also, have you tested if these effects are autography dependent? Do you need me to ask again the first part of the question?
Yes, please.
Yeah, it was a tough one. So, have you done RNA expression to try to identify an expression of signature of ICD? And then, hold on. I can't see all the questions when they're long, so I have to scroll to see every bit of it. Also, have you tested this? Have you tested this effect? Are autography dependent?
So we haven't performed RNA expression. We really wanted to look at the proteins themselves because that's what is more important in the immunogenic setup, and that would drive the activation of immune cells like DCs and T cells. Are these effects transposable?
Do you want me to read it through again?
Sorry, I'm answering for.
Have you tested if these effects are autophagy dependent?
Autophagy dependent. We tried doing some of these assays. We haven't yet a complete assay to propose in a screening platform format.
Okay. I think that I read all the questions that came up.
Okay.
If there is other one coming up, or if there is one that we haven't answered, we'll make sure to get back to you by email. That's for sure. And also, you all have Akansha's contact details. So if you have questions coming up later on, feel free to email the questions. Akansha, I'm sure you'll be very happy to answer them, right?
Of course.
All right, so the presentation will be sent to you very shortly, and you will be able to watch all the presentation in streaming. And we will also I also have shared already the presentation in PDF format for you to go back to the data and read through with all the time you need. Thank you very much, Akanshka. Thank you very much, everyone.
Thank you.
We have another question.
And I wish you a great day. Bye-bye.
Sorry.
It seems that we have another question.
Another question came up. Do you see it, Akanshka? Do you see it?
Yes, just give me a second. Is it possible to measure any biomarkers, calreticulin, HMGB1, etc., in vivo? We haven't looked at these markers in vivo. What we look at is immune cell function and cytokines. So that would be very interesting to test. And I think in the future experiments, we should try to look at some of these by isolating tumor-specific cells. Yes, thank you for the suggestion.
All right. Okay. Any other questions? We will make sure that you get an answer by email. If any other ones come up, that's for sure. We would thank you, everyone. Thank you, Akanshka. Thank you all for coming, and have a very nice end of the day. Bye-bye.
Thank you, Krista. Thanks, everyone.